So hello, so today we are going to learn about replica plate technique. So today's our topic is replica plate technique. So what is replica plate? Replica plate means creating a replica of a plate, a culture plate. So as we know, we if we take a culture plate and we made a replica of it, we can say that the culture plate from which we make a replica, the culture plate will known as master plate, and the replica plate will known as replica plate. So this technique by which each colony is inoculated into another plate according to the numbered scheme. So the new colonies will grow on the replica plate in a same numbered scheme as it was earlier in a master plate. This method allows each clone to be tested by a variety of methods while retaining the master plate from which the clones can be picked. And this technique was developed by Joshua and Ishmael Leverberg in 1952 to provide direct evidence for existence and pre-existence mutation originated spontaneously in a microorganism. So why we use replica plate? Replica plate technique is usually used for detection and isolation of mutant. So if you want to isolate or detect mutants, you can use replica plate technique for isolation and detection of mutant. So usually it is used for detection of antibiotic resistant mutants, auxotrophic mutants, and temperature sensitive mutants. So here we have taken example of temperature sensitive mutants. So if if we want to isolate temperature sensitive mutant, it involves forming colonies at 30 degrees centigrade, then transferring the colonies on two plates, one incubated at 30 degrees centigrade and another at 42 degrees centigrade. The colony which grow at 30 degrees centigrade and absent at 42 degrees centigrade are certainly consist of temperature sensitive mutation. It means the colony which does not grow at 42 degrees centigrade is sensitive to the temperature, but the colony which grow at 42 degrees centigrade and 30 degrees centigrade are, we can say that, temperature sensitive mutants. The mutants are no longer sensitive to the temperature. We can say it temperature sensitive mutant. And so what is principle of replica plate technique? So when the replicas were grown, they were compared to the location of colonies on the master plate, and any resistant colony that appeared at the same position on the replica plate were marked. The area of master plate correspond to the mark area was cut out, and the bacteria on it is resuspended in a liquid medium for further study. So by which we can isolate mutants from the master plate. So what if I want to perform a replica plate? What I will require? So here are some requirements for replica plate technique. Sterile nutrient agar plate, which is also known as a master plate. Bacterial culture, which is sensitive to antibiotics. So here we are going to identify the antibiotic resistant mutant. For that we are going to use antibiotic sensitive bacterial culture. Okay. So, and necessary glassware, sterile nutrient agar plate with antibiotic, sterile velvet cloth containing replica block. So how to perform replica plate technique? At first we will dilute the culture and spread the cells on the surface of nutrient agar plate called master plate. So here we are preparing master plate. Then we will incubate at 37 degrees centigrade for 24 hours to develop visible colonies on the surface of agar and master plate. So once the master plate is ready, now we will invert and gently press the velvet block on the sterile master plate. Since the fibers of velvet act as a fine inoculating needles and some cells from each colony of master plate stick to the velvet. Obviously, when we gently press the velvet block on the sterile master plate, the some cells from the colony will stick to the inoculating needle type of velvet. As you have already seen that velvet has kind of thin fibers which works as a needle, and inoculating needle works as, and each cell from the each colony is stick to the that velvet. And here we can see in this diagram. So when we invert the master plate on the velvet block, then we have imprints of all colonies here, and then we press those that block on the ampicillin containing nutrient agar plate, and another which lacks ampicillin, which is antibiotic, which means without antibiotic nutrient agar plate and antibiotic containing agar plate. And here we can see that there are some ampicillin sensitive colonies which haven't grown on the ampicillin containing plate, and there are some mutants which are antibiotic resistance, that's why they grow on the ampicillin containing nutrient agar plate. So here take petri dish called replica plate containing nutrient agar with antibiotic. Invert replica plate, gently press over the velvet, stamping the bacteria cells into the surface of nutrient agar with antibiotic. Orient the replica plate identically at the application on the velvet plate. Inspect the mark, place on its the rim so that colonies that appear on a replica plate after incubation occupy the position congruent to those sibling on the master plate. After sufficient incubation, observe the colonies that develop on the nutrient medium of master plate and fail to develop on a nutrient agar with the antibiotic. Mark such colonies on the master plate and isolate for further studies. It represents mutants for a specific antibiotic. Here we have seen ampicillin resistant bacteria in a previous example. Here we have taken penicillin instead of ampicillin. Here you can see the master plate. This is master plate. This is sterilized velveteen cloth with a wood holder. These are some another type of velvet blocks or other blocks which also used for replica plate technique. So what are the applications of replica plate technique? Replica plate is used to reproduce identical and spatial colonies pattern in a new plate. It is also used for isolation and generic variant and antibiotic resistant. It can also done by comparing master or a primary plate with a secondary one. In a classification of colonies on a differing from each other in the number of known nutritional requirement, which we say as auxotropic mutant, for determination of auxotropic mutant. So here you must have a question, what is auxotropic mutants? So the mutants which are unable to grow in absence of certain nutrients are known as auxotropic mutants. So here as we used ampicillin containing plate, we can use media which lacks some nutrients, so by which we can identify auxotropic mutants. Replica plate technique is used to differentiate spontaneous mutants. Normal population detect auxotropic mutants. I have explained you before. It is useful for screening and selecting mutants. Obviously, it is used for isolation and selection of and detection of mutants. So this is all about replica plate techniques. Thank you.